In video number 8, I talked about the CBIS method.
It was a good way to produce antibodies against membrane proteins. Cultured cells with antigenic proteins on their surface are used to immunize mice. And then, the mouse immune system recognizes the extracellular domains of the antigen in its non-denatured state and produces antibodies.
Right, it makes it easier to produce antibodies for flow cytometry and antibody therapy. Another advantage of the CBIS method is that it applies to producing cancer-specific antibody CasMabs. Several of the CasMabs introduced so far were made using the CBIS method.
Oh, really?
The key is, what cultured cells are used to express antigen proteins? Cultured cells are cells taken from the body and maintained in an artificial environment, in order to study the phenomena and their mechanisms occurring in cells. Typically, cultured cells cease to proliferate after dozens of cell divisions.
Cells seem to have a proliferation limiter. So, do we need to take out cells for every experiment?
That wouldn't be easy, so scientists established immortalized cells for their research.
Immortalized cells.
Immortalized cells are cells whose proliferation limiter has been removed for some reason, and they can grow semi-permanently.
Then, we can do as many experiments using cells as we like at any time.
One way to obtain immortalized cells is to isolate cancer cells.
I see, cancers are naturally occurring immortalized cells.
The first immortalized cultured cells were HeLa cells, established in the 1950s from cervical cancer in an American woman. Since then, many kinds of immortalized cultured cells have been established from various cancers. Cells taken from non-cancerous tissues were also immortalized by genetically modifying them or using genes from viruses involved in tumor development. Nowadays, cultured cells often mean immortalized cultured cells obtained in these ways. Now, do you remember what CasMabs are?
Something in the antigen protein differs between normal and cancer cells, and CasMabs recognize the difference. So, CasMabs bind to only cancer cells.
Right. If we aim to create such antibodies artificially, we will have to reproduce the cancer-specific difference of the antigen, and have mice's immune system recognize it.
I found out. We can do the CBIS method with cancer-derived cultured cells.
That's right. Suppose antigen protein is expressed on cancer-derived cultured cells and some protein molecules are in a cancer-specific state. In that case, mice's immune system will produce polyclonal antibodies, including CasMabs. If only this is successful, we'll just have to select the B cells that make the CasMabs as hybridoma by using flow cytometry screening. Antigen proteins are expressed in normal tissue-derived and cancer-derived cultured cells, to find antibodies that bind only to cancer-derived cells.
I wonder if it's impossible to produce CasMabs using a recombinant protein or synthetic peptide for immunization.
It would depend on whether such methods could mimic cancer-specific states of antigen proteins. For example, synthetic peptides might work for mimicking post-translational modifications occurring at specific sites, such as the glycosylation of Podoplanin, described in video number 6. However, the cancer-specific distortion of local conformation, which we saw in the HER2 protein in video number 1, would be hard to reproduce using recombinant proteins or synthetic peptides.
I see, the CBIS method makes it possible, so it's powerful.
The most critical point of using CBIS is that it enables the production of CasMabs even without knowing what the actual cancer-specific state is.
What does that mean?
When we want to reproduce an antigen protein on cancer cells using recombinant proteins or synthetic peptides, we must know what in the protein differs between normal and cancer cells.
Yes, we must, of course.
But we need not with CBIS.
Yes, indeed. Cancer-derived cultured cells do everything.
In fact, in the case of HER2, the CasMab was established first, and then it was found that the HER2 protein on cancer cells is partially distorted, and the CasMab recognizes the distortion. We can establish CasMabs before identifying the cancer-specific state of the antigen. That's the main point of using CBIS.
Amazing. The enormous power of CBIS.
So now, we are talking about the CasMab against podoplanin established by the CBIS method.
I remember I learnt it before. Are you talking about the same one?
Today's CasMab was obtained from another hybridoma clone. That is, today's one and the previous one recognize distinct epitopes. We've discussed previously that the podoplanin protein has numerous glycosylation modifications in its extracellular region.
One of those glycosylations appears only in podoplanin on cancer cells, which the CasMab recognizes.
However, the previous CasMab does not seem to bind to the very site of glycosylation. It's considered so because the previous one doesn't bind to synthetic peptids including the glycosylation. Presumably, the glycosylation leads to a local conformational change that occurs a short distance away, which the CasMab recognizes. On the other hand, today's CasMab binds to synthetic peptids with glycosylation.
Then, its epitope is the glycosylation site.
Yes, the two CasMabs recognize the same cancer-specific glycosylation, but in a bit different ways. Today's topic is the study of CasMab against squamous cell carcinomas of the lung.
Today's CasMab doesn't work against brain tumors.
I didn't mean so. Of course, we need detailed studies before concluding, but at least we can expect an anti-cancer effect on podoplanin-expressing cancers if only the antibody can bind to podoplanin and induce cytotoxicity.
I see.
Squamous epithelium is the tissue on the surface of the skin, oral cavity, trachea, and other areas in contact with the outside of the body. Squamous epithelial cells are sheet-like and cover the outermost surface without gaps, creating a boundary between the outside and inside of the body. Squamous cell carcinoma is a cancerous transformation of basal cells, in a layer far from the surface. Like the brain tumors previously described, squamous cell carcinomas also express large amounts of podoplanin. One of them is squamous cell carcinomas of the lung. Lung cancers have a very high incidence, affecting more than 100,000 people in Japan every year. As a result, the number of deaths is also very high, and lung cancer patients account for the highest number of cancer deaths.
They're dangerous in a different aspect from pancreatic cancers or brain tumors.
Lung cancers are classified into two categories: small cell lung cancers and non-small cell lung cancers. Small cell lung cancers, accounting for about 15% of all lung cancers, are characterized by rapid growth and a tendency to metastasize. Most lung cancers are non-small cell lung cancers, and are further categorized into three according to the tissue of origin. The most common type is adenocarcinoma, which occurs in the lung field and accounts for about 50% of all lung cancers. The second is squamous cell carcinoma, which forms at the pulmonary hilum, the entrance to the lungs from the trachea, and accounts for around 30%. Those judged not to belong to any of these categories are called large cell carcinomas. Squamous cell carcinomas and small cell carcinomas, that occur in the lung hilum, are considered to be closely related to smoking habits.
I see.
As I mentioned, the anti-podoplanin CasMabs were established using CBIS, and today's CasMab is from clone 117.
Not the same as the previous one.
First, binding to cancer-derived cultured cells was investigated by flow cytometry. These are cells derived from lung cancer. And these are brain tumor cells.
The CasMab binds to cancer cells.
These are brain tumor cells that don't express podoplanin.
No binding at all. This means that the antibody properly recognizes podoplanin. Because it's a CasMab, it should not bind to normal cells.
Of course, that was also confirmed. These are immortalized epithelial cells, established from various tissues.
The CasMab's red and black peaks are overlapped. It doesn't bind to normal cells at all. The biggest problem is its anti-cancer effect.
This is the result of the inhibitory effect on lung cancer cell growth.
CasMab inhibited the cancer cell growth.
Yes, we may expect the anti-podoplanin CasMab to have an anti-cancer effect against lung cancer. In this video, we discussed how the cancer-specific antibody CasMabs are produced.
Even if we don't know what differs in the antigen on cancer cells, the CBIS method can still produce CasMabs.
Right. CBIS will contribute considerably to developing therapeutic antibodies for cancer. The anti-podoplanin CasMabs, established by CBIS, have shown growth inhibitory effects on squamous cell carcinoma of the lung, as well as on brain tumors. We are now developing antibody-drug conjugates and chimeric antigen receptor T cells, using the anti-podoplanin CasMabs.
I believe that CasMabs, made by CBIS, will improve lung cancer treatment and help many patients.